Alrighty, so yeah, I guess we'll get started. So hi everyone, and welcome to our first VMOL seminar of the fall quarter. So today we will be doing an intro into metabolomics. We'll also be doing a recap since we've been doing these seminars for, I guess, a few quarters now. And so we'll talk a bit about what we've learned in the past, but I think there are many of you who are new to VMOL seminars this quarter. And so we'll actually be referencing some of the videos from past sessions, and these are all going to be available on YouTube. So I'd encourage you, if you're new, to go back to that YouTube channel and watch some of the previous seminars that we'll be referencing. And then finally, we'll be providing an outlook on what we'll be covering both this quarter and then in the quarters to come, since I think we have a lot of exciting material to get through. Okay, but before we dive into some of the metabolomics and some of the science, for those of you who are new, and I guess as a reminder to those of you who have been here before, I just wanted to briefly summarize what VMOL is and what are the goals of this initiative. So as described here, VMOL basically aims to connect computational biologists with chemists and data scientists from around the globe to make sort of a virtual lab. And by doing this, we really hope to remove some of the physical, economical, and other barriers of entry into this field of science. We think that one of the really exciting things about metabolomics is the fact that, a lot of it is just interacting with data. And so we can do this virtually. And I think we've been pretty successful at this in the past year. So VMOL was started about a year ago. And since we started it, we have a Slack channel that facilitates communication across countries, across research groups, across research disciplines. And that's really cool. We can do really cool and fun science that way by connecting a diverse group of people like you all. And then we've showcased some of the work that we've done at various symposiums throughout the year. So we've been having essentially one each semester. The last one was this spring symposium in which we talked about some of the work that we've done on GNPS2, different environmental samples, and then a new stats workflow. And we'll be discussing a bit about the stats workflow later today. Okay. The other thing that we do is we host this mass spec seminar. And so we're glad you all are here. Tell your friends about it. Because basically the goal of this seminar series is to take people from zero, maybe you know nothing about metabolomics, and bring you up to really having a pretty deep understanding of different mass spec and metabolomics and proteomics too, data analysis, tasks, pipelines, etc. And so this seminar is held bi-weekly every other Tuesday, except in the summer, and it's remotely on Zoom so that it's very accessible. And moreover, all of the sessions will be recorded, like this one right now, and uploaded to YouTube. And so just a bit about the makeup of those attending this seminar, because well, maybe you're curious about where you fit in. So of those people that signed up to attend the seminar, here's the makeup. Most people are intermediate mass spec users. And so we try to provide some of the basics, but also really we have an eye towards going to advanced technologies so that we bring all of these intermediates and the no experienced users up into this advanced and expert level. So we'll see how that pie chart maybe starts to shift as we continue through this seminar series. Additionally, there are students and researchers from all levels, primarily concentrated in PhD and postdoc levels, but there's a bit of, everyone. So that's pretty cool as well. And then finally, the research disciplines span a wide range: metabolomics, natural products discovery, proteomics, lipidomics, exposomics, et cetera. So lots of omics backgrounds, but yeah, really concentrated in those who are interested in metabolomics, specifically non-targeted, and that's really what we're focusing the bulk of this quarter on. So I think that's maybe good news for you. Okay, so now let's get into some of the basics that we covered last quarter and some of the outlook moving forward. And before we dive in to that, I wanted to just remind you all of the central dogma of biology, right? And so here, when we look at an organism or even building up to an ecosystem, there's a genome, right? And the genome is the static picture of this organism, for example, right? But then the genome is transcribed into RNA. RNA is translated then into proteins, and we can look at the proteins, all of the proteins, by doing proteomics, right? But then these proteins can also work together, for example, to biosynthesize different natural products or small molecules. And we can look at all of these small molecules using metabolomics, and specifically untargeted metabolomics, if we really want to look at everything. And the exciting thing is that we can look at the metabolome of an organism, for example, but we can also zoom even further out to look at the metametabolome of a whole ecosystem, right? And so if we're looking at an ecosystem, this might include many, many metabolomes. And so we're really interested in metabolomics because it provides a really dynamic picture of what's going on, right? When we get these transcriptional and translational changes, then the result is changes in the metabolome. And so we see great temporal changes in the metabolome. And so the metabolome is basically fits together into other omics technologies like the genome, transcriptome, proteome, read out by genomics, transcriptomics, and proteomics. Metabolome is, the last piece in the puzzle. And by looking at all of these various omics technologies, we can really get a full picture of the phenotype of some organism, or in this case, some nice ocean ecosystem. And the exciting thing for us is that metabolomics is sort of like the new kid on the block, right? Genomics, transcriptomics, and proteomics have all been around for a while. Metabolomics has too, but it's one of the newest of these omics technologies. And because of that, I think there's a lot of exciting space for innovation. And that's one reason that we're excited to involve all of you guys and sort of teach some of you who are newer about all of these recent innovations. Okay, but before we can really innovate, we have to understand the basics, right? And so today we'll be going through some advanced metabolomics tools. And so we thought that we could lay this out by looking at a pipeline, right? And so the first part of the pipeline, of course, in performing advanced metabolomics is just running the mass spec, okay? And so it's what we typically do in our labs, my lab and Daniel's lab, for example, is we run LC, tandem MS, and data-dependent acquisition. And so now let's talk a bit about that. So on a very basic level, for those of you with no experience, since there are some of you apparently from the survey, mass spectrometry is simply the science of displaying spectra of molecular masses of individual compounds and ions. Okay, and so we can think of mass spec as simply being a very fancy and very expensive way to weigh molecules, such as this one, which is domoic acid. It has a mass to charge ratio of 312.1441. Okay, and so from that exact mass, we can also understand molecular formula, etc. And so to learn more about the fundamentals of mass spec, I direct you to YouTube, since we did a whole lecture on this last quarter. Okay, now when we're doing an LC-MS experiment, the first thing we have to do is ionize our compounds, okay? And so there are various different strategies for doing this. And this, the strategy that you choose to use depends upon both the molecular weight that you're interested in and the polarity of the molecules that you'll be analyzing, right? And so we have ESI, electrospray ionization, and multi, but then we also have various other strategies like APCI, so atmospheric pressure, chemical ionization, a PPI, atmospheric pressure photoionization, and then EI, electron ionization. Okay, and these various different ionization modes all have their place. All right, but we typically use ESI in my lab and in Daniel's lab. And so here is just a depiction of how ESI works. Again, brief overview. We have our big droplets. The droplets become smaller, then they burst apart once they reach the Rayleigh limit. And then we actually have our charged ions, and those can go into the mass spec. Whoops. But, you know, very brief 2-second overview. We describe this in a lot of detail, again, in that mass spec fundamentals video on YouTube. So I encourage you to go watch that one. All right. But how do we actually get these molecules to the ESI? Well, that's where chromatography comes into place. And so again, in Daniel and my lab, we often use liquid chromatography, HPLC or UPLC, and that is described in detail, again, at this link on YouTube, where we give a detailed description of HPLC and various stationary phases, including C18, C8, phenyl, etc. What's the difference between these? Well, we go through all of that on in the YouTube video. But then this quarter, since this is a class or a seminar in advanced techniques, we'll be going beyond liquid chromatography and thinking about, well, what about ions interacting in the gas phase? This is where ion mobility comes in. And we're really excited because on November 7th, in a few weeks, we'll be having a seminar with Aron Baker, and she's really one of the pioneers in the field of ion mobility. She'll be telling us about why ion mobility can be really useful in metabolomics when some of us typically just think about using LC. So we're really excited for that. Now, after chromatography and ionization, then of course we have to actually do the mass spectrometry. And so we can use a variety of different types of analyzers. We went through that in another seminar, and that again is on YouTube. Here we see a TOFF with a little depiction of the internal components of that TOFF. And so again, just very quick overview. How does a TOFF actually work? Well, we have some packet of ions. Those can be separated out. And then we would see that as an MS1. But then we can actually take a mass of interest and break that with a hammer, fragment it into its pieces. And now we actually can get an MS2 or a tandem MS spectrum of that molecule. And this tandem MS can act as a sort of fingerprint for our molecule of interest, like domoic acid, for example, which can be broken where the arrows indicate. Okay, and so we can actually then use this tandem MS in some more advanced workflows that Daniel will be describing in just a minute or two. But how do we actually get all of these different tandem MS spectra? Because I just showed you one molecule fragmented, but in a real experiment, you know, we have thousands of different compounds and we'll fragment many of them. But how do we choose which ones to fragment? So again, in Daniel and my labs, we typically use DDA, which is data dependent acquisition. And here's a little picture of how that looks. We do one MS1 scan, and then we basically tell the instrument we want to fragment the top 5 ions in that scan or the top 3 ions in that scan. And you can see that's actually what's going on here. Okay, so we do one MS1, and then from that data, we figure out which top 5 ions to actually fragment, breaking them with the hammer like I showed previously. And this is what it looks like actually on the mass spec. But then we're super excited because in addition to this DDA-based approach, right, where we do a survey scan, we select top 5 precursors and then fragment those precursors, we can actually do a survey scan and fragment all of the precursors in each of the colored windows. And that's called data independent acquisition, right? Because we're not relying on the data to tell us which compounds to fragment. We're simply fragmenting compounds in each isolation window. And so we have a seminar in 2 weeks. I think that's actually the next one. Or maybe there's one in between, but we have a seminar on data independent based metabolomics. And in the past, we actually explored data independent acquisition for high throughput proteomics. And so you can watch that to kind of get up to speed on DIA. And then please come to this DIA based metabolomics. It should be very exciting. And again, we have sort of a leader in the field talking about that as well. So that is all for my part. I'm gonna turn the stage over to Daniel, but I hope that I've started getting you excited about what's to come.
Awesome. So much. Thank you so much, Alegra, for this great intro on especially the hardware and like data acquisition part. So now, yeah, like the other part of the seminar series obviously deals with the data and particularly some state-of-the-art annotation and yeah, like statistical tools that we might want to focus on. And I think one of like the mainstays for us has been molecular networking. So just very brief, you can either watch like the recaps on YouTube of those things, but what we use mainly in the lab is feature-based molecular networking, or now an advanced version of it, ion identity molecular networking, where we structure all these MS-MS spectra that we generate in this data independent or data dependent acquisition, and then yeah, like connect them based on like their MS-MS similarity to then display them in yeah, like these molecular networks. And in case you wonder how we exactly do that, so we use like the cosine similarity metric, which basically compares like 2 spectra to each other and has like this modification awareness. So we basically allow mass offsets between like the fragments here to occur depending on the mass difference of like 2 spectra to compare on like the precursor level. So if you compare 2 spectra that like differ by, for example, a precursor mass of 14, you would also allow these offsets to occur here for some of like the fragment ions. And then what you would hope is to generate, yeah, like this modification aware MS-MS similarity, where we can like connect nodes from like different molecules that share like most of the pattern, but have like a modification. Such in this case here, a modification of like one of the carboxylic acids from the moic acid, which then would correspond to the offset of 14. And yeah, if you look at like the possible combinations of like the elements, then this could obviously be a methyl group then, which happens to be also a frequent biochemical modification. So now, yeah, besides like this connectivity on like the MS-MS level, as I mentioned, the yeah, like advanced version of this ion identity molecular networking does not only look at yeah, the MS-MS spectra, but also assesses like the coelution profiles on the LC level, where we can then also create connectivities within these networks based on coelution profiles, such as we typically observe them for like in-source fragments or like different ion adducts. So in this case, like sodium or even like some dimers of the molecule. And yeah, as I mentioned, this was talked about extensively, both by Tomasz Pluskal as well as like Robin Schmidt, who actually programmed most of the ion identity and now also MZ-Mine part. With a great hands-on tutorial about MZ-Mine 3. So please check this out if that sounds interesting to you. So then, yeah, what we can do from there is obviously propagate annotations throughout this molecular networks. And yeah, this is a, I think, very beautiful slide Robin generated where it showcases some of like, yeah, like this expansions of like this spectral coverage, where we can then find new analogs. And for people in the natural product field, this is of course very, very useful, because we can like rapidly propagate annotations throughout these networks, and also like putatively identify new compounds, new natural products, or metabolites. So yeah, I think molecular networking is like kind of like the mainstay of our current pipeline we have in the lab. And I think many of you may use very similar pipelines, where we first do feature finding with MZ-MIND3, and then use GMPS2 now, as well as like Sirius for annotation. And one key thing has also been to like kind of like make this even more simple. I mean, you may know like MZ-MIND3 is a beautiful like graphical user interface based software tool that you can download and install locally on your computer. And then GMPS you can run in the cloud. And Sirius is kind of like a hybrid. You can like run it like with like a GUI, but then it needs to connect to the server in Jena. And I think all these tools are like very powerful, but also complicated. So a key motivation has been to also like rapidly or like massively simplify them by actually generating like this like completely browser-based tool, such as like the GMPS dashboard, that kind of like combines like the analysis. So you can do visualization, feature finding, and then directly launch your molecular networking job from there. And I just want to highlight the URL. So if you want to find out a little bit more of like these different tools and possibilities, then please take a look. There have been also, as I mentioned, extensive seminars before, both from Tomasz and Robin on like the feature finding side. Then myself, I did like a one on like the feature-based molecular networking. And then I think Kai Dürkop from the Bücker Lab did also like a hands-on tutorial on the use of Sirius. So now, we get from feature finding to annotation, and then we get like this massive feature table. So one big effort in the lab, and I particularly want to point out Abser and Axel there, who spearheaded that, was to streamline the statistics tools that we use downstream. And I'm very much looking forward to actually have them present here in a couple of weeks, our Hitchhiker's Guide to the statistical analysis of feature-based molecular networks, where they will present our streamlined workflow both in Jupyter notebooks as well as like Colab environments for, depending on your gusto, R notebooks or Python notebooks or Chime2 notebooks, and also like a very simplified web application in case you don't want to do any scripting and just want to click through a web app. So please join us on October 10th when they're gonna walk us through there. And then, we now can like make use of these tools to analyze our own data. But I'm also very much looking forward to actually have Ming here to talk a little bit more about like repository scale analysis. So there have been like multiple tools emerging over the last couple of years that like facilitate not only the analysis of your own data, but also the reanalysis of other public data and the contextualization. MAST would be one such example. And I think in December, Ming is gonna present a little bit more about MassQL. This is like the latest development here that allows like customizable searches of MS spectra and MS1 spectra within your own data, but also against like the full repository. And then, I think within that framework, I'm very much looking forward to actually next year have that we have a kind of like an entire series where we will present the new iteration of those tools within the GMPS2 platform, which is just launched at the moment, and that there's already some beta versions for some of them out. So this was for the LCMS part, but obviously, as Alegra mentioned, there's much more technologies than just like ESI-based LC-MS-MS. In particular, for like the use of like spatial metabolomics, there have been tremendous developments in the field, and now you can also do MS-MS in some of like those spatial metabolomics experiments, MALDI-based spatial metabolomics experiments. So I'm very much looking forward to have Stefan Heukeroth give a seminar on spatial metabolomics, and particularly the use of MALDI MS-MS-based imaging. So yeah, in case you're not fully familiar with it, what we do in MALDI is we do not separate things over chromatography, but rather shoot directly with a laser on a matrix-coated surface of a sample to then ionize the molecules. And yeah, we can raster like certain like grids where like a sample is spotted. This can be, I don't know, like a tissue section, but it can also be a kind of like some agar sections for like visualizing bacterial interaction on like agar plates. And yeah, as I mentioned, I'm very much looking forward to have Stefan here presenting on that. And then, yeah, last but not least, we also want to talk a little bit more about metabolomics beyond only metabolite inventories. So typically what most of like these experiments do that we described is that they basically describe like the chemotype of a given sample, right? And in a standard metabolomics experiment, you will have typically different groups that you want to compare. And if you have like a knockout versus a wild type, you can then like distinguish which signals or like metabolites may be missing, and then derive some like biological hypothesis out of this. But yeah, like I think a focus in the field and in many different labs, and also in my own lab, is to kind of like, in addition to like this pure inventories, also provide some functional information. And yeah, like there's been a lot of like guilt by association and correlation-based approaches where people use like genome-wide knockout libraries or so to like systematically assess the connection between genes and like metabolites. But we also want to go beyond that and actually measure directly biophysical interactions or activity relations to distinct metabolites. So yeah, I'm looking forward to both like Alegra and actually my own seminar, where we'll talk a little bit more about like native metabolomics. So these are technologies that we developed together. And yeah, like Alegra is gonna focus on like metal metabolite interactions in the realm of like siderophores. And I myself gonna focus a little bit more on like metabolite protein interactions. And then last but not least, we also want to talk about reactivity and like synthesizing actually metabolite libraries. And there I'm very much looking forward to have Emily Gentry talking about reversed metabolomics. This is, I think, one of her big projects that she did during her postdoc in Peter's lab and now in her own lab, where she synthesizes like this libraries of new metabolites and then uses like repository scale tools to actually match like those MS spectra to then like real biological data to like find out which one of those are actually real. And I don't know if you noticed that, but I recently got quite a boost in some annotation rates where now my experimental data actually matches to the spectra Emily provided. So I'm very thankful for that, and yeah, like looking forward to have her here in December. All right, so I think this is pretty much like the preview of the upcoming seminars we're gonna have. Again, so yeah, we were doing this since a while now, so there have been a lot of like topics talked about. So please, that document I shared with you in my email yesterday, check it out. What is there? There's like this past seminar list, and like there's direct links to YouTube, so you can like watch it if you want, or you can also browse our YouTube channel and see there's also some recordings from the summer schools we did, where we even talk more in detail about each of those aspects. If you've not signed up, please sign up to like the mailing list so we can like email you the Zoom information before the seminar. And yeah, I think with that, that's pretty much it for today. I think we have quite some time left. So if you want to stay on and actually ask a couple of questions, we will be happy to answer that. But yeah, roughly that's it for today. I'm looking forward to the seminar and hope to see you all around again.